Hello, everyone, and welcome to today's keynote webinar titled Understanding the Contents of Cannabis Products, Proposing a Universal Taxonomy System. This webinar is a part of the 7th annual event in the Cannabis Sciences virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I would like to remind everyone that this event is interactive. We encourage you first to participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the bottom left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button from the lobby within the event. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits image located on the right of your screen and follow the process to obtain your credits. Now let's get started. I'd like to now present today's speaker, Dr. Jokūbas Žiburkus, Associate Professor, University of Houston, Neuroscience, cannabinoid expert, entrepreneur, and innovator. For a complete biography on our speaker, please find it on the left of your screen. Jokūbas, thank you for joining us. You may now begin your presentation.
Good morning, everyone. It is an absolute pleasure to be a part of this event. Thank you, LabRoots, for inviting me and for putting a tremendous program for the audience today. I'm humbled to lay the groundwork and introduce the subject matter on cannabis and cannabinoids as the first keynote. And I think that everyone that will participate in this event will draw a lot of interesting and new information and interesting, provoking thoughts as well. So let's begin. First of all, when we speak about cannabis and cannabinoids, I'd like to walk everybody through a few slides that explain some of the major differences, because those are very important things. The devil here is in the details. So let's begin. The classification of cannabis or descriptions of cannabis. Typically, when we think of hemp or industrial hemp, it is considered a low THC cannabis plant. Hemp and marijuana are all cannabis plants. Marijuana is in fact a derogatory word, and cannabis, low THC cannabis for hemp or high THC cannabis for medicinal or recreational cannabis are more applicable terms. Although there's nothing that does not make low THC cannabis and hemp products also medicinal. In general, cannabis sativa plants are industrial hemp plants, and typically the percentage cutoff for THC, delta-9 THC, the intoxicating phytocannabinoid, in these plants are at 0.3%. And that is cutoff that is designed by the regulatory agencies. And this 0.3% limit for THC varies across the world, where, for example, in Switzerland, it's 1% THC that still qualifies the plants as hemp plants. Those plants, though, are typically, originally we are grown for medicinal uses, for fiber uses, and for nutritional uses, as well as building homes and producing a number of different products. Medical and recreational cannabis, sativa, cannabis sativa, cannabis indica, mostly actually hybrids that are available currently on the market are bred in a different way. They're typically shorter and are bred to produce high concentrations of THC or other cannabinoids, including CBD. And on the right, we have synthetic, what I call street cannabinoids and marijuana, which is not cannabis and it's dangerous. Now, I want to preface this, that this slide was made 10 years ago. And when I made the slide 10 years ago, under the synthetic and fake and dangerous synthetic cannabinoids, there was really just things to worry about in what we call the head shops, such as Kush and Spice, that were synthetic cannabinoids that were sprinkled over the plant matter, often tobacco or something else. And we're very hard to track for the regulators, and different formulas were constantly being developed. And yes, they were being peddled not only in the head shops, but also on the streets, causing deaths in many instances because they are synthetic. They're not the same acting properties. They don't have the same acting properties as phytocannabinoids. Now, 10 years later, today, I will actually talk about semi-synthetic and synthetic cannabinoids that are now in part derived from hemp and part derived from cannabinoids that come from cannabis plants, although they still present a lot of unknowns and potential dangers as well. But this is something that we will be discussing. In general, to understand this, if you see this slide, the low THC, this is the hemp, this is the marijuana plants or high THC cannabis, and this is what 10 years ago synthetic cannabinoids looked like. Now, cannabis is a complex plant, and cannabinoids are produced on the trichomes, on the tips of the trichomes, these translucent looking structures that are typically densely covering the flowers of the cannabis plant. And cannabis plant synthesizes cannabinoids. It synthesizes terpenes such as monoterpenes and sesquiterpenes. It also synthesizes lignans and flavonoids. And it's not only the cannabinoids that have the medicinal properties, but also terpenes as well have medicinal properties, and flavonoids and other elements found in cannabis plant have medicinal properties. In fact, even seeds that produce high levels of omega-3 and omega-6 acids are very valuable health-wise, not just nutrition-wise. For major cannabinoids, when we talk about major cannabinoids, there's CBGA, THCA, CBDA. Another example is CBCA, and we'll look over these. For monoterpenes, it's limonene and myrcene. Sesquiterpenes, beta-caryophyllene examples, beta-amarin. Lignans are cannabis and D, flavonoids, canflavin A, some of the examples. They all have their own precursors at the bottom here that I've summarized from this reference article here from 2016. All have their precursors. They all have distinct biosynthetic pathways that lead to the synthesis of cannabinoids, terpenes, and other molecules shown here. Cannabis plants within these trichomes synthesize acidic cannabinoids. So cannabis plant in its raw form does not produce CBD or CBG or THC. In fact, it synthesizes the acidic versions of these cannabinoids, CBDA, CBGA, THCA, CBGA, or cannabigerolic acid, becomes a precursor to THCA, which is tetrahydrocannabinolic acid, and also becomes a precursor to CBDA, and that is what is produced in the plant. And currently, from all of the cannabinoids, these are just the major cannabinoids, but in fact, there are over 100 different phytocannabinoids that have been described, even more so than 100. There's only 10% CBD formulation that is accepted pharmaceutical by FDA, and synthetic delta-9 THC tablets. And delta-9 becomes important here. So the plant synthesizes delta-9 THCA. It has this carboxyl group here attached, a OH group right here. And once it gets decarboxylated, it becomes delta-9 THC, a neutral version of that cannabinoid. CBDA, once it gets decarboxylated, shown here, G, will become CBD. However, currently, we also have on the market such things as delta-8 THC and delta-10 THC, and they're quite often sold as naturally occurring, although there's a debate, and I would argue that they're not naturally occurring. They're byproducts of extraction or heating processes or other chemical alterations from these other natural phytocannabinoids like delta-9 THCA or CBDA. The other molecule that is shown here, 11-OH-THC, is the major psychoactive metabolite that delta-9 THC turns into within our bodies, into 11-OH-THC. So this is something that is present in our bodies through the secondary metabolism also. We'll come back and discuss these synthetic cannabinoids in a few minutes. Now, besides cannabinoids, and as I mentioned, there is many different cannabinoids. If THC and CBD and CBG would be considered major cannabinoids, but because they can be produced at high concentrations or higher concentrations than other cannabinoids, there are also what are called minor cannabinoids, CBDG, CBDV, varin-like cannabinoids. And they're called minor cannabinoids because they're rare. So not many cannabis strains may produce and synthesize these minor cannabinoids. And in addition, if they do synthesize those minor cannabinoids, they will be present in very small amounts, percentage-wise, as a total per weight amount in the cannabis plant. In addition to phytocannabinoids, those translucent trichomes that we saw in this previous slide that produce and synthesize acidic cannabinoids, those translucent trichomes are also synthesizing through the biosynthetic pathways, as we saw earlier, terpenes, monoterpenes like limonene, other terpenes like caryophyllene and linalool, as shown here. Now, these terpene molecules are aromatic volatile molecules, and they're not unique to cannabis. In fact, fruit like lemons and limes and oranges are loaded with high concentrations of limonene, and caryophyllene can be found in cloves and peppers, and linalool is heavily expressed by lavender. These cannabinoids in our bodies interact with the endocannabinoid system. And this is a topic that could be discussed separately on its own in a whole separate hour, the endocannabinoid system, the endocannabinoids that are produced in our bodies, the cannabinoid receptors, CB1 and CB2 receptors that are widely expressed throughout our bodies and brains. And then the endocannabinoid molecules are essentially endogenous, our own body-produced molecules. They are major endocannabinoids in our body are anandamide, abbreviated AEA, or 2-arachidonyl glycerol, which is abbreviated as 2AG. These endocannabinoids are produced on demand by our bodies. There's certain steady state level of endocannabinoids in our bodies and brains, and virtually every cell in the body and the brain contains elements of the cannabinoid system, which is, again, synthesizing enzymes for these endogenous molecules. Those endogenous molecules, endocannabinoids, cannabinoid receptors, and potentially other receptors outside the CB1 and CB2 that are mentioned in this particular slide and image. And also the degrading enzymes that are then going to take care of the endocannabinoids. Endocannabinoid molecules are like neurotransmitters in the brain, but they're different from other neurotransmitters because they are lipid soluble, they're cellular plasma membrane soluble, and for that reason, they're not stored in vesicles like most of the neurotransmitters chemicals in our brains. Endocannabinoids production and synthesis is started by increased levels of signaling in the brain or increased levels of stress on the cells. And endocannabinoids in particular in the brain will regulate and balance the chemical neural transmission. And it will have different effects in different parts of the brain as well as different parts of the body. What this diagram shows is that the brain is dominated by CB1 receptors, and the heart is also dominated by CB1 receptors, but organs like spleen and bone marrow and organs that are associated with the immune response, and even in the brain, CB2 receptors would be associated with the immune response. They would be, again, these organs will also be dominated by CB2 receptors. There is a homeostatic regulation of endocannabinoids off the brain and the human body. Endocannabinoid levels get increased in human milk to promote newborn suckling, to promote hunger feeding and also pleasurable feelings. It gets kicked in the production with pain stress modulation, and it has many different effects: immune function, appetite, memory, inflammation, thermoregulation, and other effects. And as you can see, these endocannabinoids, like anandamide shown here, actually similar in structure to the phytocannabinoids. And the phytocannabinoids, such as THC and CBD that we were already discussing, will be also interacting with these cannabinoid receptors. So let's look at, for example, we already understand cannabis plant is pretty complex. We also understand the endocannabinoid system is really complex, and it potentially is individualized amongst all of us because we would be expressing slightly different receptors in slightly different areas of the brain or different organs, and the different ratios of these receptors, as well as the ability to synthesize endocannabinoids ourselves. Some folks can synthesize more endocannabinoids, and certain activities can produce synthesis of endocannabinoids. For example, long distance running is thought to produce high levels of endocannabinoids and cause what is called the runner, or the euphoria that many runners feel. That's from the endocannabinoids. And how do these phytocannabinoids then interact with the endogenous system components? In this case, we're going to look at just one receptor target, which is cannabinoid receptor 1, CB1 receptor. And we will look at the three major phytocannabinoids: CBGA, which is the precursor to CBDA, and THCA. The plant will make these acidic versions of cannabinoids. These acidic cannabinoids, with heat, with decarboxylation, with extraction or chemical processing, can turn into the neutral versions. THCA becomes THC, CBGA, CBG, CBDA, CBD. That's important. So the plant will contain acidic versions, but then with heating, with burning plant matter, THCA, delta-9 THCA, which is produced in the plants, becomes THC. THC is an agonist to CB1 receptor. That means an agonist, it means it's going to activate the downstream functions of this receptor, and this receptor is what is called a G protein coupled receptor. So activation of CB1 receptor by THC will set off a downstream cellular cascade and will regulate neural transmission in neurons in the brain. CBG is an antagonist to CB1 receptor. That means that it is going to inactivate the CB1 G protein coupled receptor activation, as well as inactivate the downstream cascades that otherwise this receptor would be mediating inside the cells. CBD is an allosteric modulator, a negative allosteric modulator. That means that it binds to a different site than THC that binds to this receptor. These receptors are like doors that have many different locks, and these molecules are like keys that can fit into different parts of this door where there are many different locks. And so THC will have its own slot on CB1 receptor, and CBD will have a different slot where it can bind to. And when it binds to it, it essentially dampens the activation of this G protein coupled receptor. CB1 receptors in the brain, once again, here we just looked at one target, cannabinoid receptor 1, and we already understand that just with three major phytocannabinoids, they will exert a different effect and a different cellular effect through the activation or dampening of the CB1 receptor activity. CB1 receptors in the brain are the most abundant G protein coupled receptors. This is really significant because G protein coupled receptors are involved in many different modulatory, long-lasting functions inside the cells. They're involved in modulation of learning and memory, and also formation of new connections or synapses between neurons, and strengthening of the communication between neuronal circuits. And so let's look at the CB1 receptor distribution in the brain. What we know about the brain is that brain has many, many different structures and divisions with it. Even if we look at the cortex, different parts of the cortex are processing different information. The occipital cortex in the back is primarily concerned with the visual information processing. The parietal cortex will contain somatosensory areas. The frontal cortex and prefrontal cortex here is listed as responsible for executive functions. So there is a distribution and division of labor between different parts of the brain, and then the different parts of the brain interconnect with each other and communicate this information. For example, emotional response from amygdala gets joined with the learning and memory responses from hippocampus, and that will affect the executive function and one's behavior through these interconnected neuronal loops. And what this diagram illustrates is the abundance of cannabinoid receptor 1 expression in different parts of the brain. So hippocampus is an important area for learning, memory, and modulation of stress. It's shown here in red, has high CB1 receptor expression levels. Basal ganglia, which is responsible for cognition, learning, emotions, and motor control. Cerebral cortex, which is higher cognitive functions, processing, and also modulation and also integration of different senses, such as sense of smell and hearing all at the same time. So we can see that there's very wide expression of these cannabinoid receptors. At the level of the brainstem, and that is important, at the level of the brainstem, we have some of the nuclei that control some of the vital body functions. There are nuclei in the brainstem that control sleep and wake cycles, so sleep arousal, thermoregulation or temperature, and motor control. So these are in solitary tract, nucleus of solitary tract here that is involved in visceral sensations and nausea and vomiting, and another periaqueductal gray that is involved in analgesia. So this is where you find at the level of the brainstem CB1 receptors. That is very important. And as you can see, CB1 receptors are not expressed in the parts of the brainstem that are responsible for respiration, for heart rate, or for breathing rate, and that is very important. Instead, activation of these responses, receptors in this part of the brain will have analgesic, anti-nauseating effects, as well as regulate motor control and sleep cycles at the level of the brainstem. But everywhere throughout the brain where there's going to be interactions of these molecules with CB1 receptors, there's going to be a different effect. So we're all different human beings. We have different densities of these cannabinoid receptors, and different composition of cannabinoids and different strains of cannabis plant will produce different compositions of cannabinoids, will interact with these receptors in different brain areas in the brain, thus causing fairly individualized response from cannabis. In addition to cannabinoids, I mentioned these trichomes, cannabis plants will produce terpenes. Terpenes are volatile aromatic molecules that give cannabis its smell. Terpenes, a lot of times cannabis will be described as smelling like lemons, or smelling like skunk, or smelling like gasoline, or smelling like blueberries or lavender. And those smells come from terpenes. And those smells are, if you think about it, not just purely psychological effect of when you smell things. There's actual physiological effect that happens when we smell strong odors, when we have strong odor stimulation. In this example that's taken from an article on trends in cognitive neuroscience and cognitive sciences, it shows recordings of the brain activity using fMRI, or functional magnetic resonance imaging. In this case, what you're looking is these red hot spots. These red hot spots and orange hot spots indicate areas of heightened neuronal activity and neuronal synchrony. And this illustrates that once we smell a certain odor, that odor activates, of course, our nasal epithelium and olfactory receptors and olfactory bulbs, which is located right near our nasal epithelium, near the nose. But there are very vast connections from these primary sensory olfactory neurons, smell neurons, and those projections go throughout different parts of the brain. And there's a very strong connection to an emotional center of the brain that goes from the smell centers here. And there's very strong connections throughout. So that means that when you smell a particular odor, it changes the physiology of the brain. It generates these spatiotemporal maps of neuronal activity inside the brain that can be tracked and that are actually unique in shape and in time, as is illustrated here. There's of course some similarities between mint, rose, banana, and lemon map in the brains, but there are also some distinct differences in these maps. And how our brains are activated by the smell, by olfactory physiology, is that we actually change our behavior. So this is a true physiological effect, not just through smelling things. In the case of cannabis, it's through ingestion or inhalation of terpenes that come a part of a lot of cannabis products, in particular cannabis flower. There are many medicinal properties of terpenes. They're aromatic lipophilic compounds that easily cross membranes as well as blood-brain barrier. And that's the barrier that exists between the blood and the brain. And it is a policing control station that oversees what substances can cross from the blood into the brain. And these molecules, limonene, myrcene, pinene, that are listed, that have their own medicinal properties, these molecules can easily cross through blood-brain barrier. And these are just some of the examples of the medicinal properties of terpenes. And here's another slide that there's some interesting articles that are quite recent that came out that are discussing the synthesis and synergy of these terpenes in cannabis sativa. Some of these terpenes in cannabis sativa and cannabis plants are cannabimimetic. That means that they will mimic the activity of cannabinoids, and in some instances will selectively enhance cannabinoid activity. There are also recent studies that show that some of the cannabinoid activity, like from THC, because THC can evoke anxiety, some of the terpenes can actually counteract this anxiolytic or pro-anxiolytic activity. So there are different interactions with terpenes and cannabinoids where in some instances they can enhance cannabinoid activity, in some other instances they can dampen cannabinoid activity in the brain. And then there's this other interesting article, Medicinal Properties of Terpenes Found in Cannabis Sativa and Humulus lupulus. And Humulus lupulus are hops, because cannabis and hops are the closest related plants on this earth. This is a very interesting medicinal properties of cannabinoids, terpenes, and flavonoids. So I haven't talked much about flavonoids, but obviously these other molecules that are synthesized by cannabis plant have their own medicinal effects. And I encourage you to look up these references on the slide. And there's a lot of information, and how do you sort through all of this information? You sort through this information by trying to systemize things, by trying to create some sort of a system, some sort of a taxonomy. In this case, this is an attempt to have a taxonomy of medicinal properties of terpenes, where they're subdivided into monoterpenes, for example, sesquiterpenes, polyterpenes, and all of triterpenes, and all of these different terpene groups are being targeted, for example, control of apoptosis, or acting as anti-metastatic, anti-inflammatory, or in this case as anti-angiogenic. So this is interesting because this is, to my knowledge, kind of the first attempt to take large groups, not necessarily individual terpenes even, but large groups based on the structure, and assign those groups to specific conditions that are indicated here. Despite the extensive medicinal use of cannabis, dating in written records to 5,000 years ago to Emperor Shenlong's records in current day China, there's very limited availability of cannabinoid pharmaceutical medications. And this table illustrates what cannabinoid pharmaceuticals are actually available to patients by prescription federally in the US and also outside of US. Raphael Mechoulam shown here on the left, rest in peace. He passed last year. He isolated and correctly described the structure of THC in 1964. And only in 1981, there was a synthetic delta-9 THC tablet, Nabilone, and in 85, another delta-9 synthetic THC tablet, dronabinol, containing 2.5 milligrams of THC. It's used as appetite stimulant, antiemetic, typically in terminally ill patients or patients that are undergoing chemotherapy. This was not an original idea by the pharmaceutical companies. What pharmaceutical companies observed is in the 1970s, there was a significant development and innovation going on in cannabis. A lot of it had to do with the draft dodgers in the West Coast running to British Columbia to avoid the draft. In fact, creating the famous BC bud and breeding a lot of different cannabis plants and also using the cannabis and extracting cannabis and making preparations and oils and extracts from cannabis. And it was noticed in the 70s in the so-called West Coast hippie community that people that were undergoing through wasting syndrome because they were compromised with, their immune systems were compromised with HIV and they had AIDS. Those people did not have any appetite. These patients were wasting away and they were dying. And when they were exposed to cannabis, it would stimulate their appetite and something that is known for a very long time. And it would allow them to eat and it would also lessen their pain at the same time. So this was something that was happening in the society broadly, especially on the West Coast, that pharmaceutical companies observed and said, we'll just make a synthetic THC and put it through safety and efficacy studies. That's what the pharmaceutical is. It's approved for safe and efficacious use for specific conditions. Between 1985 and 2005, there is no other medication from cannabis that is being accepted in the international community. In 2005, there's development of nabiximols, which is plant-based, the first plant-based, and it's not FDA approved. It is available in over 25 different countries, but it is not FDA approved. And it is a combination of delta-9 THC and CBD, 1-to-1. It's a buccal spray, a cheek spray, where we have a lot of absorption through the mucosal membranes. It is antispastic and analgesic, and it is being used in multiple sclerosis. But again, this preparation is not available in the US. In US, first starting in Europe and later in the US in 2018, we have cannabidiol. It's 10% CBD oral solution or tincture or drops essentially that is anticonvulsant to stop seizures in Dravet syndrome, a severe myoclonic epilepsy of infancy, Lennox-Gastaut epilepsies, and also infantile spasms. And at first it was placed on Schedule 5. Now it has been actually descheduled. And this is the only plant-derived phytocannabinoid pharmaceutical FDA-approved drug that we have, 10% CBD oral solution. That's it. So we need a way more because we have a lot of availability of cannabis. And this is what's really confusing to the consumers and to the healthcare professionals because the laws are changing all of the time. And this illustrates the map from 2022, I believe, where some of the states will have adult, robust adult, and medical use regulated programs. Some of the states will not have any access still, a handful, 2 maybe of these, and Then there are states that will have CBD, low THC programs, but those programs are also now being overcrowded in a way with the hemp-derived cannabinoids. And in Texas, for example, there's over 2 billion in sales of hemp-derived cannabinoids. Most of them are synthetic or semi-synthetic. And the THC program, marijuana program, that otherwise would be regulated with medical supervision is completely stagnant. So this is the effect of this improper really use of different cannabinoids in unregulated markets, that they can impede the progress of regulated market. They can impede the progress in health, and they can essentially be harmful in many aspects because there's no standardized ways of producing these different molecules that we have. It's also confusing because there are different global legal cannabis THC systems. So we just talked about Marinol, Sativex, the pharmaceuticals that are national, that are health insurance subsidized. There's also national medical cannabis programs in Canada and in Germany, and they're health insurance subsidized also. There are state-regulated programs. An example is Massachusetts, which has robust adult use and robust medical use program. The only subsidy there is that medical patients are exempt of the 20% tax that the recreational users or adult users have to pay. But there's still no health insurance subsidies because it is regulated at the state level. But I think there's an opportunity potentially to change that over the next few years to a decade. There are different state cannabis programs that are adult use or recreational, that are state regulated or nationally regulated. In Canada, these recreational adult use programs fall under full taxation, and quite often province or state regulated in the United States. On a national level, Germany has joined this list together with Canada to become a country, largest country in Europe to legalize adult use cannabis. Now, when a consumer looks at this map, they have to be careful where they are at the moment. And if they're traveling for work, they have to remember, and sometimes laws change so fast and they're not even aware or not sure what the local laws might be at the moment. And when somebody goes and purchases cannabis products as a consumer, or even as another business, all cannabis products are labeled differently. They're often impossible to understand. So for example, it's this little layout here, and it's very difficult to understand what concentration of THC. It takes time to study this label, and that becomes an impediment. So cannabis plant is complex enough. The products that get produced from that plant can be even more complex in the composition of the active ingredients, and the labels can be overwhelming and very confusing. And in the end, a lot of consumers will rely on the names of the products or the names of the strains, which is really just a story. It really quite often does not have to do with replicable active ingredient composition, even in the same titled products or the same strain name products. That leaves consumers very confused in order to select the right products, to have the right medicinal effect, or not to overdo something to have a good experience. In general, I put the blame squarely back on the cannabis industry. There's a lack of science and research on the products in the cannabis industry by companies that are profitable in some instances already, that are publicly traded, that circle budgets of tens of millions of dollars for marketing, but fail for the most part to validate any medicinal claims or produce significant research on the actual products. And if they do, it's often self-reported rather than empirical data that comes from these studies. Furthermore, there's no standard. There's no national standard. There's no international standard. There's no standard in the state. And your most trusted advisor becomes a person behind the counter who may not also understand even the level of the science that we're talking about here today. So when we look, for example, at these major cannabinoids, we have delta-9 THCA and delta-9 THC comes from the plant. CBDA, CBD comes from the plant. Delta-8 gets synthesized from CBD. So there is no delta-8 in the plants, and you'll see that these semi-synthetic or synthetic cannabinoids like delta-8, delta-10 in the states that do not regulate what are called hemp-derived cannabinoids will have advertisements all over every head shop, gas station, hemp stores. In fact, I did some research by walking into the hemp store and I couldn't find phytocannabinoid products. Every product was either contaminated or was purely a synthetic cannabinoid product. Delta-8, delta-10, HHC, which is a hexahydrocannabinol, THCP, which is tetrahydrocannabinol, full roll that has this very long tail of carbon chains here attached to the rings. And when you walk into a lot of the stores, there's a variety of products, a variety of labels. And this is an example of some of the products that are available in the state of Texas and available online that are selling delta-8, that are selling delta-9, that are selling HHC, that are selling THCP, synthetic cannabinoids that are listed here with detectable values. And this is extremely confusing to everyone. So to solve this problem, we need to start a taxonomy system. There's been an attempt to have a taxonomy system for the plants, cannabis plants divided into cannabis sativa, cannabis indica, and cannabis ruderalis, mostly based on the morphology of the leaves, how they appear, longer sativas versus shorter, bushier indicas, and also geography. Most of the plants in reality are hybrids. There's very rare what we call landrace original strains that will be sativa or indica. Most of the plants are hybrids. And there's another attempt to then do a cannabis plant taxonomy, THC dominant type 1, THC and CBD balance type 2, and type 3 CBD dominant. And this is a very broad description of still the plant taxonomy based on purely THC and CBD dominance. There's other attempts now. So besides trying to have a taxonomy for cannabis plant, we need a taxonomy for cannabis products. This is a taxonomy, that visual representation of strains that was created by Leafly. It's flower strain system that represents cannabinoids and terpenes. So cannabinoids are presented in different shapes, diamonds or pearls, and terpenes in different colors. It's only applicable to strains, and it's quite difficult for the consumers to remember. And it is also based on the average certificate of analysis or average chemical results for that particular named strain. And what we have to realize is that same strain named from the same seed can be grown on the East Coast and the West Coast, and they will contain a completely different, not completely different, but very significantly different chemical output in as far as the levels of THC and CBD, and also terpenes as in color here. True Terpenes and others have produced these color systems for the terpenes. There is a major international standardization body, ASTM, that is working on standardizing the processes, standardizing even the labeling of cannabinoids. This is a very welcome initiative that we highly support, myself personally too. And this is the first sign that has been produced, a cannabinoid warning sign. Now we need a cannabinoid synthetic warning sign. That is a synthetic cannabinoid that the consumers can be aware of. While the regulators are going to catch up with the laws, we can be conscious at informing the consumers what they're consuming. Need for universal cannabis product taxonomy. We have major product categories: flowers, edibles, tinctures, concentrates. Major categories have different percentages. Cannabis flower cannot produce typically more than 36, 37% of cannabinoids by weight. Edibles, so quite often they can be 5 milligrams a dose or up to 100 milligrams. Tinctures, they can be a 100 milligram bottle of cannabinoid, sometimes 5,000 milligram a bottle of cannabinoids in different volumes. Concentrates can reach up to 100% if it's a pure isolate. And so we need to standardize and create a taxonomy for these products. Uniwyze has attempted to do that where they have placed all of the products in the strength categories for each type of product. So whether it's flower or concentrate, based on these concentrations or edible, it becomes a light, moderate, strong, and extra strong. And it is based on science, it is based on what is available in nature and what is possible to produce in these existing products. Yes, this might be an arbitrary attempt to produce this type of a taxonomy that is broad and universal for all cannabis products, but it is a necessary attempt to fix the current market problem. We have strength categories, need a simplified visual representation rather than complex visual representations. There has to be a warning on synthetic cannabinoids. We must start thinking about producing an active ingredient score that is not based on the marketing, but is based on what is in the finished product, based on the certificates of analysis. What are the active ingredients in there? What are their attributes of these active ingredients? And to get rid or reduce the use of subjective descriptions and subjective properties and subjective recommendations for different types of products. Because cannabis sativa and cannabis indica may contain the exact same amount of major cannabinoids, THC and CBD, and the exact same major terpenes. And so to say that sativa will do this or indica will do this is inappropriate because they can contain the same active ingredients in the finished product. So we have to take away the subjectivity, introduce a classification system based on strength and an ingredient score system across all of the products. And once again, if we look at what is existent here, there's a lot of subjective claims, companies that will make a lot of subjective claims, companies that will not have simple visual representation, that will not categorize products on strength, but will ask you to choose what percentage you would like. They will not have an ingredient score, but rather a score of how consumer likes the product or some other subjective measure. And none of these systems so far in the market, except maybe with the exception of one or two, are applicable to all of the formats of products. So once we have in place a simple visualization system, and that is based on the taxonomy, that is based on the empirical objective data, and that avoids subjective recommendations, I think we can improve the market and we can improve experiences for both the producers, manufacturers, retailers, as well healthcare practitioners, which could be helped a lot with that, and finally the consumers of this product. So I hope that you found this presentation useful and we're able to gather these major learning objectives of understanding the complexity of the plant, recognizing the current cannabinoid challenges in the market that contain a lot of unvalidated claims, subjective claims, and synthetic cannabinoids, and that hopefully you start developing appreciation for the need for standardized cannabis system and standardized cannabis practices. I will be happy to take any questions from the audience.
All right. Well, thank you so much, Jokūbas, for your informative presentation. We will now start the live Q&A portion of the webinar. So to our audience, if you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, so let's get started. We've already got some great questions coming in here. This first one I have for you asks, what is the role of CB1 and other cannabinoid receptors in the brain?
So this is a very good question because during my presentation, I focused primarily on CB1 receptor, or cannabinoid receptor 1. And cannabinoid receptor 1, CB1 receptor, is predominantly expressed on neurons in the brain. Neurons in the brain are responsible for chemical synaptic transmission. And these CB1 receptors are located mostly on the presynaptic endings or nerve terminals of neurons. And when these CB1 receptors get activated by endocannabinoids or phytocannabinoids, the CB1 receptors will decrease the release of neurotransmitters from the presynaptic terminals, and they will regulate the release of glutamate, which is a major excitatory neurotransmitter, and they will regulate the release of GABA, which is the major inhibitory neurotransmitter in the brain. So CB1 receptors on neurons, in that case, when there's heightened activity between neuronal networks, endocannabinoids would get produced on demand, and they will target CB1 receptors through what we call retrograde signaling, and will regulate neurotransmitter release. CB2 receptors is the second cannabinoid receptor that is most abundant in the brain, and CB2 receptors are predominantly expressed in the cells that are called glial cells. So whilst neurons are responsible for synaptic communication, this chemical communication, and CB1 receptor can affect this chemical communication, CB2 receptors are expressed on the cells called glial cells. And those glial cells are responsible for different functions from neurons. In particular, CB2 receptors in glial cells will regulate the release of pro-inflammatory cytokines. They will regulate the inflammatory and also the immune response of the brain cells, following infection, or damage. And these are slower processes. So CB1 will affect more faster neural transmission. CB2 will regulate more of the homeostasis of these neurotransmitters, as well as inflammation and immune response in the brain. And throughout the body, we could go down in every organ, but I think we will probably give opportunity for other questions and just focus on the brain with this answer.
Great, thank you. All right, I have another question for you here. What about HPLC or TLC fingerprinting analysis and standardization of cannabis products?
That is a very good question as well. High performance liquid chromatography, it's the technique that allowed, originally it came about in the, again, 60s and 70s, which is why the cannabinoid drug development is also tied to about the same timeline. HPLC allows us to isolate individual active ingredients in cannabis and describe essentially the chemical profile of the active ingredients, even in cannabis flower or cannabis products. And I think that this is exactly the correct way to create a taxonomy and to categorize all of the cannabis products. Because if we do it by strain name, as I explained, one of the popular strains, for example, is Blue Dream. And if you're in California, you purchase Blue Dream and it had a certain concentration of THC and CBD, and then you moved to New York State. And then in New York State, you walked in the shop and you said, I want Blue Dream. And somebody says, oh, sure, I have Blue Dream. But a person will not realize that that same name strain, and even sometimes the same name product, will contain a different composition of cannabinoids. Therefore, they will not have the same experience, whether it's medicinal experience or relaxation experience with that same product. And what HPLC fingerprinting would do, essentially, it would allow for us to track all of the active ingredients in the finished products and create a system, a categorization and visualization system based on the certificates of analysis. And so this is a very important thing. There's also another question that goes along with that. Somebody that is into genetics would say, what about the phylogenetic profiling of cannabis plants and strains and taxonomy? That is great, again, for plants. But I think we have to go to analytical methods and standardization of this, analytical methods and standards for analysis. And as we have the standardization of analytical methods, also should go into categorization and standardization of all of the cannabis products that are based on the final active ingredient composition as illustrated by these analytical test results. I hope that answers the question.
Great, thank you. Now it looks like we have time for one more question, and this question does have two parts to it. Concerns about hemp-derived intoxicating cannabinoids are valid, But are there any inherent negative health impacts of consuming them when processed responsibly? And what processing or manufacturing concerns do you have about these products?
That is a great question. And I do realize that there are producers of these products that are quite responsible in tracking and tracing the origin of all of the ingredients, have very clean labs that are GMP certified labs to produce all of the ingredients and produce all of the products. However, I think that that is just a small portion of what is happening in the market. So if we did have a responsible, regulated pathway with track and trace for some of these synthetic or semi-synthetic cannabinoids, I'm not saying that they should be banned. The concerns that I have is that there's a lot of unknowns. People have been using Delta-9 THC and CBD for millennia. There have been many studies that have been done with these molecules, but molecules like HHC, THCP, there is no long-term knowledge of what they can potentially do. So we have to be careful. We have to be careful, and I would encourage those same responsible manufacturers of semi-synthetic or synthetic cannabinoids that are making billions in revenue, up to 30 billion potentially last year, according to Whitney Economics. I would encourage those responsible operators to be concerned about what their consumers are consuming and to validate and to do health research and development of their products. So you can play a part in actually showing that some of these cannabinoids produced in a safe way that you or somebody else produces can have a beneficial effect rather than just an anecdotal effect. And so that would also be helpful. I'm not a proponent of banning things, but I'm a proponent of communicating very clearly what those things are and their origin. And somebody will say, well, what is wrong with synthetics? You take synthetic medications, there's synthetic caffeine being sold everywhere around the stores. It's, again, not the question whether it's semi-synthetic or synthetic, it's how it is being presented. This comes from the plant. I can walk into many stores and ask Delta-8, and they will show me 20% Delta-8 result on the flower and say, this comes from the plant. It's in the plant, and we know from scientific method that is just not possible unless this should go in the Guinness World Records book right now. So those are some of the concerns that I have. I would promote the responsible production and marketing and very translucent, transparent communication with the consumers, and also be concerned about the fact that if these synthetic and semi-synthetic cannabinoids, now you can look at the list of them being tested, like up to 40 or something like that in some of the labs, they will potentially have a chance to destroy the whole medical and potential adult use national system, or at least slow it down. So we should all work together, and we should help each other to promote responsible production and labeling and transparency of these products.
Great. Well, thank you, Jokūbas. That will conclude our session for today. Do you have any final comments for our audience?
No, thank you very much for very good questions, for being here this morning, and look forward to participating in this event, and hopefully you will continue and stay on as well. And if you want to contact me, please feel free to use the Gmail that is listed on the slide. Thank you very much for the organizers one more time.
And thank you again, Dr. Jokūbas Žiburkus, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 17th, 2025. LabRoots will alert you via email when it's available for replay, so we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Goodbye.